Healthy skin is not just a matter of appearance, but something centrally important for human health. It is our first-line defense against the outside world. It is what we see when we look at other people and what we feel when we touch them. But sometimes it can harbor life-threatening disease, a melanoma. Melanomas are malignant tumors originating from the degeneration of melanocytes, the cells that produce the pigment that gives our skin color. In Switzerland, there are over 12,000 new cases of melanoma every year. Risk of developing the disease is high among those with a pale skin tone, like the majority of the Swiss population, as it offers little protection against dangerous UV solar radiation. The national passion for outdoor activities doesn't improve the situation. Olivier Michielin is leading a Systemsx.ch research project in Lausanne, aimed at revealing the different ways metastatic melanomas adapt to current therapies and limit their long-term efficacy.
Metastatic melanoma is a very aggressive disease. With current treatment options, 20% of the patient can be stabilized and doing well without symptoms for at least 10 years. With newer regimens, we are moving to 40%, and with current research, we hope to move the bar to 80, hopefully 100% in the coming years.
Up to the end of the last century, tumors were mainly classified and treated according to the principal organ involved: breast, prostate, lungs, and so on. But over the last 15 years, it has become more and more clear that each tumor is a highly complex agglomeration of different cells, not all of them tumorous. To some extent, every tumor is unique. This complexity explains why the same therapy does not always give the same result in two different patients.
In order to improve the number of patients who can benefit from treatment, we need to be able to understand the disease in molecular detail. We need to be able to understand why the disease in a given patient is responding, whereas the same disease in another patient does not respond to treatment. We also need to be able to understand why a disease is responding initially in a patient and then is progressing. So we need to be able to analyze the molecular pattern, the mechanism by which the disease is now able to resist therapy, to bring further discovery and new therapeutic solution for the patients.
Olivier Michielin and his collaborators study samples, each containing about 1,000 cells.
We analyze gene used by single cell obtained from the tumor. We can analyze the tumor before treatment or after treatment. We see very big differences in the gene used before and after, which help us design subsequent therapy.
In other words, the cellular composition of a tumor changes under therapy. Some dangerous cells are usually killed, but in many patients, some of them survive or even go on to develop resistance.
With this project, we are learning a lot about melanoma. This is research at the moment, but each time we understand a new mechanism, we are getting closer to personalizing the treatment for that patient.
In the fight against cancer, scientists are increasingly able to confront each individual tumor with a combination of different strategies that include these standard ones, such as chemotherapy, but also the more advanced ones, such as immunotherapy, which are increasingly targeted to hit each cell's Achilles heel. This is a goal that could never have been reached without a systems approach to biology.